again this Wednesday, July 27th, 2022. I'm glad I caught that now. And I'm joined by Mike Stone from Virology OG, Mike Donio, our good friend, and Dr. Andy Kaufman. And without any further introductions, the reason what we're talking about today is a few weeks ago, we put out a viral challenge, which is this. And you can find this. Tricia will put the links so you can read and find the document. And it gives a definition of what a virus is: a small parasite consisting of nucleic acid, RNA, or DNA enclosed in a protein coat that can replicate only in a susceptible host cell, and I would add, and by doing so causes disease. So the reason we did this is to basically try to ascertain whether virology is actually a scientific enterprise, or whether it's based on false premises and experiments that are actually invalid. And we are, of course, questioning the entire existence of pathogenic viruses. And obviously, if they don't exist, they can't cause disease. And so this whole COVID situation is based on science or anti-science and things that are simply not true. Now, as a result of putting out this challenge, which is for me very gratifying, we've had a number of people who've responded to us saying, well, here is a paper that shows, quote, irrefutable evidence that the virus exists and it has a certain sequence and causes disease. And then another doctor, I may be a virologist, I'm not sure, made a video showing us photographs of the virus, claiming that this proves that the virus obviously exists and is doing what they say it does. So that's, again, what I had hoped would happen. So hopefully there's no name calling, there's no bitterness, there's simply just a statement of a paper, a statement that the paper, a publication of a paper, the paper is supposedly irrefutable evidence. And so we thought we would take a look at that paper and those electron microscope pictures to see if in fact they are irrefutable evidence or not. So that is the point of today. Now, the first paper, again, let me show you the paper. Just to be clear, this is a preprint, so it hasn't actually been published to my knowledge yet. I'm pretty sure that's accurate. So here's the paper we're talking about. And the author, and it looks like the only author, is a man named Sin Lee. And I don't want to make a mistake in his credentials, but I believe he's a medical doctor and a pathologist and a virologist. But I could be wrong about that. But that's my impression. Now, Andy and I have actually had an encounter with, let me call him Dr. Lee, before. It was probably over a year ago when we were invited, I was invited to present to a bunch of lawyers, Kennedy's group and Fulmick's group, and other people about the idea that there's no virus, and therefore SARS-CoV-2 doesn't exist and cannot cause COVID. And they were, I would say, not thrilled with my saying this. And so as a result, not with my knowledge, I would say, but invited Dr. Lee to rebut what I was saying. So that was my first and prior to this only encounter with Dr. Lee. So I asked Andy to join me, and graciously he did. And that was, I think, really important. And so we gave our presentation, things that most of you, I'm sure, have heard. Interestingly, then Dr. Lee had his turn, and he didn't actually respond to any of our comments or actual questions or information or facts. Instead, he did three things. Well, he did two things that, in his mind, I think, proved the existence of the virus. The first was he showed us a picture of a young Chinese fellow on a respirator. I don't know where the picture was taken. I believe in China, maybe even Wuhan. And so we saw, without any other information, a picture of a young fellow on a respirator. And he said, this proves there was a virus causing his disease. Now, whenever I hear things like that, I look for what is the principle underlying this statement. And what I would say about that is that the principle is there is such a thing as clinical virology. In other words, you can determine whether the cause of somebody's illness is a virus by looking at their symptoms. And we hear this a lot. My 3 golfing partners got sick, and then I got sick. It must be a virus. People in San Quentin were sick, so it must be a virus. My Aunt Bessie went to church, and she got sick, and it must be a virus. Lots of people in Wuhan got sick, and it must be a virus. The principle, again, is if a lot of people have the same symptoms, that proves it's a virus. Obviously means things like Hiroshima must have been a virus because a lot of people in the same place got sick. But the other point here is there is no clinical virology. You can't, as we were told, say, well, what about rabies? In other words, people get sick in a certain way, maybe from a bite, maybe, and that proves it's a virus. The reason for that is human beings, all of them that I've ever met, have no awareness of a virus without doing laboratory experiments, period. You have no feeling there's a virus. You have no perception there's a virus. There's nothing in your experiential world that tells you there's a virus. Now, let's compare that to something else. You could say, well, molybdenum deficiency causes a certain set of symptoms. Now, again, nobody I know has a sense of whether you have molybdenum in your body or not. Obviously, you can't sense that. You can't feel it. The only way you could know that is if somebody does a very careful experiment and shows that these symptoms can only be because of a molybdenum deficiency. After that, if that was proven, then you could say if somebody has those symptoms, it's from molybdenum deficiency. But before doing that, that is simply nonsense. And so anybody telling you that a picture of somebody who's sick without having proven the virus exists and causes this disease, that is irrational, illogical, and anti-scientific. Now, so that was the first thing. The second thing is he said, well, I purchased a vial of pure SARS-CoV-2 from this store, which I think was an online virus store. And so I don't remember which one of us asked him, how did they make that pure virus? Obviously, if you say this is a pure virus, you would know how they came up with that product. So unfortunately, he didn't know. Now, I'm sure most of you could guess that this was not a purified virus. It was not purified SARS-CoV-2. It was it was a toxic cell culture that had broken down, and they claimed this contained this SARS-CoV-2 virus without any proof that this was actually the case. So I think this vial cost a lot of money, and frankly, I don't think it was worth it. So again, this is not proof, and the fact that he didn't know how you came up with this virus, with this product, is definitely disconcerting. Now, the third thing, and I'll say about this in a minute, we asked him, is it true that there is no published reports of anybody finding any pathogenic virus, including SARS-CoV-2, directly in the fluid, biological fluid of any sick person? And he said, correct. And we asked him, why not? Because obviously the human lungs, in the case of COVID and SARS-CoV-2, is the natural culture medium for this virus, allegedly. Therefore, if you say the person is teeming with virus, you should be able to find it in an electron microscope. He said you couldn't. And then I think it was Andy had the presence of mind to say, well, what about if you took 10 people and collected them together and took their bronchial fluid? Would there then be enough virus to be able to find? And he said, no, not enough virus to find. What about 100 people? No. What about 1,000 people? No. What about 10,000 people? If all of who you say have SARS-CoV-2 have COVID, you supposedly demonstrate that with whatever test, is there enough virus to find? And he said, no, there's not enough to find, and then he wouldn't answer any more questions. Now, this is the reason why I say fundamentally virology is an art, because I wouldn't call it a science, of saying things which, if you understood what they were saying in a normal situation, you would never believe. And just a quick example, if you came home and the wood was rotting in your house and you said it was being, I have a theory that it's being eaten by vermites, and the vermite exterminator people come and say, the vermites are eating your house. And you say, did you see the vermites at my house? No. Have you ever seen vermites at my house? No. How many people's rotting houses have you examined? 10,000 people. Have you ever seen vermites? No. So what makes you think there are vermites there? Well, we found a piece of a wing there, and we analyzed the molecular composition of this piece of a wing, and we said that must be vermites. Now, think about this for a minute. If you've never seen a vermite, how would you know that that piece of a wing came from a vermite and not a termite? I believe termites have wings. Or a fly, or a mosquito, or a bird. They all have wings, and they all have probably similar molecules, and there is no way to know. If you want to know the genetic material of your cat, you take the cat and you take cells or tissue from the cat and you find out what it's made of. Now, this paper that we're going to examine, and Mike and Andy will examine it in a little more detail, not only is not irrefutable evidence that there's a virus, there is no evidence that there's a virus, mostly because, well, there's no virus, but the other reason is because they didn't even look for a virus. They never made any attempt to do what virologists are meant to do, which is, find the virus in a sample, or since they can't do that, culture it, prove that only the virus is causing the CPE, show us purified particles, take the material, the proteins and the genetic material from those particles, and prove those sequences came from those particles. Absent that, just like the vermites, lots of things have genetic material, lots of things have wings. You have no idea whether there's those sequences, which is the entire rest of the paper, where they came from. And so with that brief introduction, let me turn this over first to Mike. and go into a little more detail into maybe what I just said, or actually what they did in this paper, or what he did. And thanks all three of you again for joining me, and thanks for listening. And yeah, Mike, take it away.
Thank you, Tom. So I'm not going to go into too much detail because I know Dr. Kaufman and Mike are going to be breaking down more of the finer details of the paper. But I just wanted to point out some of the things that concern me as far as when someone puts a paper such as Dr. Lee's paper as irrefutable evidence. What we're seeing with virology is that they're trying to move us into this molecular virology where they don't need, or they claim they do not need, the purified isolated viral particles in order to prove the existence of a virus. All they need is their sequencing data. So that's pretty much what we see with Dr. Lee's paper. Is it's nothing but genomic data. There's no attempt at purifying and isolating a virus in any sense of the word. There's no electron microscopy images of the particles that they claim are to be the virus. There was no attempt at establishing pathogenicity in a natural way with either animals or humans. They didn't even try, replicating the results or reproducing them. I mean, it's still under, I believe, under peer review at the moment. Take that for what it's worth. But we see this trend where they're moving away from like the old ways of virology, saying culturing a virus is not necessary anymore. It's too time consuming or too expensive, or you need all this equipment. So why do that? Why not just go with a sequencing? Let's get a genome, and that's the proof of the virus. But what I find interesting is back in 2000, I believe it was 2001, virologist Charles Kalischer and a group of, I believe it was 13 other virologists, ended up warning the new guard of virology that they are relying too much on this their new molecular toys, is kind of how they put it. And he basically said, these strings of letters, the DNAs, the ATCGs, in a data bank tell you absolutely nothing. It can't tell you, how a virus replicates. It can't tell you how it, infects people. It can't tell you if there's any antibodies or anything that react to it. It's basically, he put it, it's like trying to tell someone that they have bad breath by looking at their fingerprints. So the thing is, they're trying to claim that they don't need to purify and isolate a virus. All we have to do for this irrefutable evidence is to look at this paper that is sequencing data. So my question is, how is this paper irrefutable evidence? If I were to go and try to claim that a virus exists, especially in the case of SARS-CoV-2, I would look at the foundational papers, the ones that came before. So, January 2020 through March 2020, when they were trying to make the case that this virus exists. So why are we looking at a paper that is now 2.5 years into this as irrefutable evidence when we're not supplying the foundational papers? And I believe that they are doing this primarily because they admit that the particles, or the virus particles, have never been purified. They've never been isolated. And so it's easier for them to kind of wiggle through this using genomic data. And so, we can go into each of those foundational papers, like the Fan Wu paper that had the genome, the Zhao, the Zhu papers that admitted to not purifying, or at least not fulfilling Koch's postulates. They didn't, I believe the Zhu paper admitted to not purifying their EM images, things along those lines. But to me, this is the more concerning issue, is that they're moving away, even though, our challenge is to try and verify if the methods of virology are actually valid. They're actually trying to move away from the cell culture and all these things and just rely primarily on genomic data to make a case that these viruses exist. And we've seen how that can lead to, you know, misusing PCR and other methods to try and tell someone who's completely healthy, they don't have symptoms, that they're sick, when in fact there is absolutely no proof that this virus exists and is actually causing people to become sick.
Okay, I think to summarize, it's basically, and I want everybody to try to answer this question for themselves. Is it possible to claim that you have proven that a piece of something comes from that something if you've never actually isolated, purified, and examined that something? Is there any other example in your real world? If you think your foot came from you, it's really helpful to know you first and that that's your foot. Okay, Mike, what can you say? Tell us more about this paper.
All right. Thanks, Tom. Yeah, I mean, I would absolutely agree with Mike that what you have here is a situation where they're making this huge claim that this is the absolute irrefutable proof. And that's a big, bold claim to make. And then you start off by not even purifying, isolating a virus. You're just starting with genetic material. And actually, it's really quite informative to just look at the methods of the paper. And that's kind of where I always go first when I'm trying to decide, you know, is the claim being made by a given paper valid, because that will tell you largely based upon what they've done, whether they can back those things up. I mean, first of all, with this paper, it was kind of interesting because normally you find kind of in the abstract or the introduction that there's some sort of a well-articulated, you know, hypothesis or an idea of what the question is that they're trying to answer. And I felt like that was kind of missing from this. So you kind of go into it a little bit unclear. And then when you look at the methods, they're kind of just starting with material from 5 people from nasopharyngeal swabs. So, you know, swabbing in the nose. And then they're not isolating any particles or making any attempt. They're saying, I believe that they confirm them to be what they call true positive, which, you know, I'm not sure what exactly that means, but it was done by a Sanger sequencing protocol. And that's how they're determining that these samples are positive. There's also no other controls. They didn't evaluate samples from healthy people or anything else to confirm, you know, that these samples were actually unique to someone who was sick and diagnosed with COVID or whatever. So you're kind of starting off on shaky ground because you don't actually have that independent variable, that isolated virus. And then, you know, as you kind of work through and you look at what they did when they isolated, so they isolated RNA, and it's kind of interesting because they say that they take these samples and dilute them, and then they, I guess, centrifuge them or something, but then they actually say that there's a cellular pellet. So they must have pelleted something out of these samples that then they go in and kind of resuspend and digest. And that's where they're pulling out the nucleic acid, the RNA. And then from there, you know, they're going through a series of steps of RT-PCR. So they're converting it from RNA into DNA. And there's a lot of problems associated with that reverse transcription procedure, not the least of which the enzyme reverse transcriptase has a very high error rate. And then you're going through and doing PCR. We know that there's plenty of issues there. And then they do a second round of PCR, and this is where they're calling this nested PCR that's supposed to make it more sensitive, but it's a little bit weird how they're doing it because they didn't actually, I don't know, it's not easy to explain, but they actually kind of amplified almost the same exact piece again. And then throughout this whole thing, then they go to the Sanger sequencing of what they're claiming to be their product of these reactions, but they never at any time clean up this reaction and even pull out a fragment that they believe is specific. They're taking the crude material that they isolate from digesting this cellular pellet from the swabs and carrying that through each step, and again right into the Sanger sequencing without purifying out. They claim that they found, I assume on doing agarose gel electrophoresis, where you can run these things out and you see these bands that correspond to size. And so they're claiming that they did that and they saw a piece that was the size they were expecting. So then they just moved along, but of course they don't show that. And then they're not cleaning anything up. They're going right into the Sanger sequencing, and then they produce the sequence data. And I mean, I've done things like this a lot, and you would never carry crude sample throughout this process and not clean it up, maybe pull out the exact fragment that you're looking to sequence and say, look, here's what it just seems like a very weird way of doing it. Especially if you're that convinced that these sequences are deriving from what you're claiming to be a virus. And then, I mean, I know Sanger sequencing is claimed to be somewhat more, I guess, less manipulable than the methods that's used to generate the genomes, because it's in those cases, you're assembling from all these small pieces, but Sanger sequencing is still limited to a very small piece, because beyond a certain size, you get a lot of errors and it gets just noisy. And they actually showed these they're called like chromatograms or whatever in this paper, where you can see kind of where they're pulling this sequence data from. And you can see some of the pieces, they're quite noisy. And so, there's a lot of issues, I guess I would just say, just looking at the methods and the data that's being presented itself. And that's, again, without even having that independent variable, that virus in the first place. So it's a really weird place that we're in when we're just assuming that we've found an organism, in this case a virus, based on identifying some random sequence without ever having observed the organism itself. And I think we're just back in that same kind of place where we're making assumptions without ever having the thing.
Mike, let me just maybe rephrase a little bit and see if this captures, and maybe Andy too, you could weigh in on it. I think one of the reasons this paper was put out now was because some of us have said that if you do isolate, purify the virus, pull out the genetic material, and then you should Sanger sequence it, which is essentially clipping it off piece by piece or nucleotide. That's more reliable. And believe me, I could do a whole thing on the problems of Sanger sequencing, but let's just say it's more reliable than taking these little pieces and putting them together like a puzzle. In other words, assembly. And so he's essentially saying, well, right, we did Sanger sequencing, so therefore we're good. But that's not actually the issue. The issue is, what are you Sanger sequencing? And as you said, and I think I got this, I'll get this right, you're Sanger sequencing little pieces of of genetic material, which you have no idea, no proof, no even attempt to prove that they came from an organism, let alone this piece of an organism. And so it doesn't matter what kind of sequencing or assembly you do at that point, it's still, that's not the problem point. It is itself a problem, which is why you keep getting different answers, therefore different variants and different mutations, et cetera. And so that's really what the issue is. I don't know if that makes sense to you.
Yeah, Tom, I think this is like an old, really scientific problem, which is when you have compromised input into a process, then your output is going to be compromised. And really, next generation sequencing would be fine if you started with one intact genome that you could go back to and check yourself to make sure that all the pieces fit together the way they did in that long strand. And or you could do micropore sequencing, or you could do Sanger, right? All of those things could be valid if your starting material is actually has a known origin.
Yeah. Okay, Andy, take it away.
Well, thank you, Tom. And I just want to follow up briefly on what Mike Stone was saying. And by the way, both of you guys really did an excellent analysis. Mike, in my opinion, and I've described this before, the move toward molecular genetics as the proof of a virus, I think is driven by the manufacturing. So just like if we go back to Enders, who developed the cell culture methods, they were based on manufacturing the polio vaccine. And now we have a shortcut to manufacture a genetic-based, gene therapy vaccine much, much quicker, because we can essentially skip all the time of growing, changing the cell conditions, waiting, passing it to various cell cultures to attenuate the vaccine, etc. The sequence gets published in a database and then can go right to pharmaceutical companies. They can, within hours, have a manufactured vaccine to start clinical trials. And so I think that's a big driving force, other than, of course, the ease of provability. So my understanding today was that we were going to, speak about the paper as people have mentioned, but that also there'd be an opportunity to ask some questions of the author. So I'm going to share my screen. I just developed a few slides here so that I could have the questions written down for whoever might be looking at this video and take up the opportunity to answer these questions. So I took a little bit of a different tact in how I looked at this paper. I essentially pretended I didn't know anything about virology and just look at it as a scientist and as someone who's familiar with logic. And interestingly, so I was told that this paper was put forth as quote unquote, incontrovertible evidence of the existence of this SARS-CoV-2 virus, but in the abstract in the title, it didn't say that this experiment proved the existence of the virus anywhere. I thought that was kind of interesting because it seems to be like a big deal that if you provide that evidence in your experiment, you'd want to say it outright at the top of the paper. But I discovered that in order to determine if this paper proves the existence of this virus, that you actually only have to read one sentence in the entire paper. Now, I did read other parts of the paper, but there's no reason to talk about it because with this one sentence, you know that the paper couldn't possibly prove the existence of a virus. And this is the first sentence, as I have outlined here, from the materials and methods section, which said that it starts out with 5 selective nasopharyngeal swab specimens collected from non-hospitalized patients with respiratory infection, which were confirmed to be true positive for SARS-CoV-2 Omicron variant. So in other words, the premise of the study is that this sample has this virus in it. Before they do any step of the experiment that they're reporting on here, they already have that premise to be the case. So you cannot use an experiment with a presupposed premise to prove the truth of the premise itself. And that is what's called circular reasoning, or begging the question, or petitio principii. And this originally is reported to come from Aristotle in the Fallacies on Logic. And this isn't a bona fide Aristotle quote, but if he were alive today, he might say something like that. But I do have a couple of scholarly quotes to read here to help you better understand what the logic fallacy is. So the fallacy of circular reasoning occurs when the conclusion of an argument is essentially the same as one of the premises of the argument. Circular reasoning is an inference drawn from a premise that includes the conclusion and used to prove the conclusion. And another way of saying that is to beg the question is to attempt to support a claim with a premise that itself restates or presupposes the claim. So in other words, you just make a claim and then restate it as the conclusion, and there you go. So turns out that is actually the entire basis of virology. And so I'm going to show you here in two of the pillars how the circular reasoning begging the question plays out. So if you look at the flow chart on the left, this is with respect to virus isolation experiments or the cell culture experiments we have discussed so far. And the premise is that viruses are present in the patient that you're doing the experiment on, and that viruses cause the so-called cytopathic effects. Then you take a sample from the patient who has the virus, you put it in a cell culture with the other additives per their recipe, then you wait, you observe the cytopathic effects, and then you conclude that it was caused by a virus that was present in the patient that you began with. So in other words, you've essentially restated the premise as your conclusion, and this is the fallacy here. Now this kind of a statement can sometimes be useful to add redundancy, an emphasis of meaning, but when it comes to drawing a conclusion with respect to discovering something new in nature, it is a pure fallacy and cannot be used. So I developed a few questions based on this particular circular reasoning argument for the author. Firstly, how do you know that the cytopathic effects in the cell culture virus isolation experiments are a direct result of a suspected virus rather than any other cause? And of course, there are many other causes that could result in that. Number two, why aren't control experiments with the same exact experimental conditions, varying the independent variable only, which is simply the virus in the specimen? So you'd have to have another lung fluid specimen without a virus or with a different virus to be a control. And why isn't that experiment routinely performed? Because it's a requirement for any scientific experiment to have a control in order to be valid in terms of proving causation or the existence of something. And then number three, why aren't virions directly extracted and purified from patient samples? If there aren't enough, how can there be enough to cause serious disease? And this is really speaking to the same question that Tom mentioned in the introduction. So I want to show you another example, and this is sort of more related to the molecular genetics that we've been discussing today of circular reasoning. And sorry, my computer here is not cooperating very well. So you can see at the top, once again, the circular diagram, and this is related to the in silico genome. And once again, I want to bring your attention that we're not talking about the current paper, because that is built already upon the assumption that the genome is of a real virus and it's a real genome. So if we're going to really look at this, we have to go back to the original genome experiment, which I think was the Wu Fan paper that Mike Stone mentioned from Nature. And that's using a technique called metagenomic sequencing. So that's what I'm talking about, and that's how you originally determine the viral genome. And the premise of that experiment is that, of course, once again, viruses are present in the patient that you're looking at, and that viruses have a genome of 30,000 base pairs. So then you take the patient sample again, and this time you extract all of the RNA from all sources, and you make what's called a complementary DNA library, and you use PCR without primers to do this because you don't know what you're looking for, so you can't develop specific primers. So you use what they call non-specific primers so that you amplify everything that's there no matter what it is. Then you sequence every single one of those strands of RNA, and in that experiment it was over 56 million. And you put them into a software program. In that case, they used two different programs, and it generated over 1 million possible or candidate genomes. And it did this by matching overlapping ends of the sequences. And then, of course, the conclusion was that they ended up picking one of those over 1 million candidates that had 30,000 base pairs, and they called it the genome of the virus. So in other words, once again, they restated the premise as their conclusion. Thus, another example of begging the question or circular reasoning. So I developed the following questions based on this line of experimentation. And once again, I want to remind that this only refers to metagenomic sequencing, so no additional subsequent sequencing protocols that were used to identify variants, for example. So question 1. How do you know that all of the oligonucleotide reads that were incorporated in the putative genome are from a virus and not any other organism from the patient sample? Number 2, how do you know the length of a genome before it's been discovered? Number 3, how do you know the other million or more contigs that are not the how do you know that the other million or more contigs that were not chosen aren't the actual correct genome of the suspected virus? Number 4, why do they have to go back at the end and do additional PCR to find the sequence of the ends of the genome? Wouldn't they be included in the original set of reads that went into the computer? And lastly, and I think we've kind of already asked this question, but why not instead simply purify the virus first, extract only RNA from the virus and not from any other organisms, and perform micropore or Sanger sequencing to determine the genome? Now, I just want to spend 1 or 2 extra minutes going over one thing that I think is something that no one is really talking about, but that we have, all of these sequences that they're looking for, that they're said to be, from a virus. And we have done various searches and found that some of these sequences actually match various microorganisms and human sequences, but not all of them match up. And we haven't really done the exercise where we've looked at all 50 million reads and tried to match them up with their origin by known sequences. But I want to say that there is an unknown variable that humans, that we and other animals also, actually generate unknown sequences all of the time. And what I'm talking about here is non-coding RNA. So when the human genome was first completed, they found that only a very tiny fraction, less than 1%, made up the genes that code for proteins. And then they had the other 99+% of DNA that they originally called junk DNA and said it had no function. But since then they learned that all of it gets transcribed into RNA, and they call it non-coding RNA because it doesn't give you a code for a specific protein. But what happens to all of that 99% plus of our DNA? So it becomes RNA, and then it undergoes further processing that actually can change the sequences. And we have kind of a meta diagram here that you can see in the middle. The blue circle is the nucleus, and you see that there's mRNA in there, that's the kind that codes for proteins. So we're not talking about that. That's in red. We're talking about everything else, which all comes from NC or non-coding RNA. And you can see that this makes a lot of different things, and this is also a very young field, so it's not even really known the extent of what happens to all of this RNA. But we know that a lot of it gets spliced into these very short little strands. And we can see in the blue section on the right, the miRNA, microRNA, for example. And those are around 22 base pairs long, which is long enough to be counted in their genome sequencing procedure. And these can undergo recombination and splicing and other types of post-translational processing that the sequences of all of this myriad of different little pieces of RNA doesn't necessarily match the DNA sequence in the chromosome. So if we tried to subtract out the genome sequence of humans from the computer database, which it's not clear that they actually do, but certainly that would be something that would make sense to do, you still wouldn't be able to capture all of the human sequences because of post-translational processing and alteration of 99% of the RNA, that it won't match what's in the genome to begin with. And of course, we know there are variations in the genome as well, which makes it more of a complicated problem. So I think a lot of what they're finding in these sequences that they're, arbitrarily calling viral in origin really are these little snippets of highly processed RNA that come from our own cells. And I will leave it there for now, Tom, but thank you.
Thanks as always, Andy. Great. So I had a series of questions for Dr. Lee, who had actually promised that, by the way, our other good friend, Mark Bailey, wrote a paper about the Sin Lee paper. And we were promised by Dr. Lee that he would specifically answer whatever questions we addressed. So rather than me showing my questions, I'll just leave Andy's questions. So if you could please address those questions and the questions that Dr. Bailey wrote in his paper. And Tricia, if you could make a link to Mark's paper, that would be great. So I think we've covered that, and we anxiously await this very clear and specific answers to Andy's series of questions by Dr. Lee. And I, as probably people know, I'm a stickler for time, so we don't have much time, but let's take a minute here to address the video that Dr. Fleming put out. And Mike, I'm not sure if you can show that video. It's about a minute long.
Yeah, let me see if I can pull it up.
Otherwise, we could link to it and let people watch it themselves if you don't have it readily available.
Okay, that's okay. It's here.
Coronavirus means it looks like a crown. This is a slice through tissue of a coronavirus. Anybody telling you they don't exist needs to look at this. Anybody who tells you that we don't have the genetic sequence, we do. This is what it looks like. These are spiked proteins, and because of these spiked proteins sitting around, it looks like a crown. And this is a cryo-electron micrograph of coronavirus.
Okay, I think that's probably good. So just to cut to the chase, can we get that?
There you go.
So, and again, Mike, see, let me just, I don't want to put words in your mouth, but basically this is saying that you can use various forms of electron microscopy, in this case, cryo, which I guess means freezing, to prove the existence of a virus, and that this SARS-CoV-2 has a very specific appearance, which we can then use this technique of electron microscopy to visualize it, and that proves the existence. Is that basically correct?
Yeah, that seems to be the premise that they're making. And yeah.
All right. So maybe people have seen this before, but I have something which I call my pumpkin challenge. And the reason I call it that is I actually now have a almost 1-year-old well, he used to be a kitten, but now he's a cat. And I taught him to say yes or no and something about virology. So I asked Pumpkin to come on the show with us, and he said he would only do it if I gave him a pound of fresh swordfish every week. And I thought that was too steep. So he's not going to appear. So you'll have to take my word for that these were Pumpkin's answers. So I showed him various pictures from the published literature that are either SARS-CoV-2, they say, or from lung biopsies or kidney biopsies from the pre-COVID era, some even before 2000. And I asked Pumpkin to say which ones were with SARS-CoV-2 and which ones were not. And he got 6 out of 11 correct. I'm not going to tell you which ones. So I'm wondering if Dr. Fleming would like to take the Pumpkin challenge. We may use different slides because you could look up these and find out which ones they are. But let me show everybody what I mean by that. So first of all, here are some quotes from the peer-reviewed literature. So this is electron microscope images in figure so-and-so do not demonstrate coronaviruses. Rather, these structures described as virus are clathrin-coated vesicles, normal subcellular organisms involved in intracellular transport. In other words, what we're saying is when you look at an electron microscope picture, and again, forgetting about all the problems with doing electron microscopy and all the processing that you need to go through to obtain this image, there is simply no way you can distinguish a, quote, virus from normal breakdown products of dead and dying cells. If you don't believe me, here's another quote. Transmission electron microscopy of tissue sections is not a specific or sensitive method for the detection of viral particles. There are numerous structures found by electron microscopy that resemble viruses, so-called viral-like particles. And they go on to say, therefore, caution is suggested when identifying a virus by EM in tissue sections, exactly what Dr. Fleming proposed to do. Here is an article from Kidney International. Transmission electron microscopy of tissue sections is not specific or sensitive method for the detection of viral particles. That's the same thing. Here's from The Lancet, apparently a peer-reviewed journal that some people read. Just recently, there have been 2 reports in which structures that are normally found in the cytoplasm of a cell have been misinterpreted as viral particles. It can be a powerful tool, but care must be taken when interpreting cytoplasmic structures to correctly identify virus particles. They're obviously hedging their bets and not willing to say at this point, there is no way you can tell the difference. And then here's from JASN, identification of viruses is not straightforward. Care should be taken to prevent mistaking cell organelles for viral particles. In other words, when a push comes to shove, you can't tell the difference between the contents of dying cells versus these so-called viruses. Most of the published images depicting the suspected virus are similar, if not identical, to multivesicular bodies. They've been known since the 60s, and we suspect that the electron microscope images of SARS-CoV-2 published to date are in fact multivesicular bodies. In other words, they're breakdown of our cells. So here is the pumpkin challenge. So this is supposedly SARS-CoV-2. You can see the arrows and you can see fuzzy spike proteins. And I won't tell you which one is this. Which one is this? Similar size. You see the black dots. Those are meant to be the spike proteins. There's many of them. Which one is this one? How about this one in the lower left-hand corner with the round structure with, again, the spike proteins, the, quote, characteristic corona Which one is this? How about this? Same picture. That one. Here you see apparently a cell with lots of these coronas inside. Is this lung biopsy? Is this kidney biopsy? Is this SARS-CoV-2? Because I can tell you, to spoiler alert, I think it's 5 of the 11 are SARS-CoV-2. The other 6 are not. Pumpkin likes to say no because he's a contrarian, so I think he mostly said no. Here's another one. Same thing. Same thing. Which one is this? Turns out you cannot tell the difference. And here's 4 different images from different papers, all showing these things, which are these electron microscope images. Which ones are these? The little, where the dots are. Are these coronaviruses? Are these multivesicular bodies? I don't think anybody could tell the difference, and we could prove that to them by showing them these blinded slides and seeing if they could, in fact, do better than Pumpkin. So I don't know if anybody has anything they want to add to this electron microscopy issue, but the bottom line is everybody, I think, would agree that you cannot use electron microscopy images and all the problems that entails to prove the existence of this particle. So anybody want to add anything to that? Andy? Mike?
I was just going to say, from the paper that they took that from, they actually, or that Fleming took that from, I believe, actually admitted to not purifying any particles. And they actually said that purification may lead to artifacts or something. Like, it is actually kind of ridiculous. So, I mean,
so this was a fake picture.
Yeah.
More or less.
Can't prove that Daffy Duck exists by showing a fake picture of Daffy Duck. That's not how logic, rational thinking, or science is meant to work. Mike or Andy, anything you want to add to this?
Who can follow up Pumpkin?
Yeah, there you go.
He's a tough act to follow, believe me. All right. I think we got it. Thanks, everybody, for joining me. We anxiously await Dr. Lee's responses to Andy's series of questions. And let's just say Dr. Fleming will then hopefully write us and say exactly how a virologist distinguishes an electron microscopy, either cryo or transmission or the old-fashioned way, through a picture with no other input, just by looking at the picture, distinguish a pathogenic virus from the breakdown contents of dead and dying cells. So again, thanks everybody for joining me. I'm not going to be doing a webinar on Wednesday, and tentatively the schedule is to address another question, which is, we were told that bacteriophages are viruses and they therefore prove the existence of the virus. And in an interesting twist, it turns out that, as far as I can tell, and I'll look into it more, there are such a thing as bacteriophages, and they actually help us prove that there is no such thing as a virus. So I will go over that next Wednesday.
I'm really relieved that you said that, Tom, because I thought we were going to have to vaccinate bacteria.
Yeah, well, there's no money in it, Andy.
That's right. Those lousy bacteria, they're deadbeats.
They're broker than Pumpkin. All right, everybody. Thanks for joining us. And Mike, and Andy, thank you as always. And we will be in touch.
Thank you, guys.
Bye, everyone.
All right.